so here is that green tree frog that Lynn just mentioned, and as I've just been having a few conversations about this, this green tree frog isn't in fact green at all. And this is one of the subjects really that I will cover in this talk. It's about the eye, about how the eye actually introduces colour to the world. And in particular, actually there's no such thing as colour. So without the eye, there would be no colour in the world at all. Actually, just on that slide moment, this is one thing that artists have been dealing with. I mean, the colour in the natural world, as I'll talk about now, involves all sorts of mechanisms to produce the optical effects. And when artists have traditionally tried to copy those, they can have fallen short because with their paint palettes, they don't have the devices available to them to recreate those natural colours. And this is one example with John Gould in the 19th century. He was trying to paint hummingbirds. He was producing these large monographs with really faithful, accurate reproductions of these birds. But he couldn't get the shimmering, iridescent colours. So he tried to do something about that, and he recognised there was a problem there. So he started to use gold leaf and silver leaf, and then he put his paint on top of that, very translucent layers, and then he used a layer of honey over the top, a glaze made of honey, and experimented with all sorts of things to try to capture that same iridescent movement of colour that you get in real hummingbirds. Because by using these paints, it was great when you looked at it from one direction, but as soon as you moved, the colours stayed the same. Whereas in the hummingbirds themselves, the colours change, and all sorts of feathers start to flash at you, and some drop out, some light up even brighter. And he just wasn't getting that with his paint. But he came quite a long way with his methods. But really, very little has been done since. And I think it's been useful to artists to understand how colour in nature is produced, to try to give you ideas about how you can make representatives on canvas that can provide a more realistic effect. And I think that's more about the capturing the parts of colour in nature that are important, because we don't use every aspect of the colour-producing structure in nature. We just use parts of it. Some parts are more important to us than others. So if we can understand that, it will help the artist to create a more faithful reproduction. So the eye itself, well, we'll begin actually with the beginning of the book set, Deadly Colours, which begins with Darwin's problem. The eye, Darwin found, was something that he couldn't conceive to have it originated from natural selection. He could understand how an ant's jaw could evolve from a leg. You can understand how you can have a leg and how you can have a series of fine changes and end up with a jaw. You know, you can see the intermediate stages and how those could work, but he couldn't see that with the eye. And one of the problems is that he thought the eye was just too sophisticated. He couldn't conceive how such a sophisticated organ could possibly evolve from gradual small steps. And this is what he wrote. In The Origin of Species in 1859, he put aside a whole section on a problem to his thesis. And he thought that, this is something I just can't explain. Because he said that he listed all the different aspects of the eye and made this such a sophisticated organ. And that just created such a problem to him that he said that the eye could be formed by natural selection seems absurd in the highest possible degree. So we're going to try to do something about that in this talk. Well, at the University of London in Sweden, Dan Erik Nilsson and his group performed this project here, which was quite useful because it shows that, in fact, an eye can evolve by a series of small steps. This shows how a piece of light-sensitive material sandwiched between a transparent lens, or so a transparent covering and a black pigment background, as you can see in the top left, how you induce a number of changes to that and gradually increase its perception to light. First of all, its sensitivity to direction. So what's happening in here in this sequence of the eye, evolution, it's starting to invaginate, it's starting to have a concavity to it. and that is beginning to increase the perception of direction. So in the first one, you can only tell if light is present or not. If it's falling from above, the light-sensitive material will be stimulated. If not, then nothing will happen. And as you start to get a more curved surface, such as there, you can start to sense which direction light is coming from. If the left-hand side lights up, then light must be coming from the right-hand side, and so on. And these numbers relate to the number of changes in terms of genetic mutations within a generation. So, within 364,000 generations, you can have the evolution of an eye from and that's in terms of most species we're talking about, less than, say, 100,000 years, which is nothing on the evolutionary timescale. There's one important thing I'm working on at the moment, and that's how you can go from this step to this one, because that's massive. What happens there, in this diagram, what Daniel Nielsen has said is fine. You've got this gradual transition, but he's overlooked the fact that between those two stages, you've got the evolution of the lens. So you go from the amount of information the retina receives suddenly leaps 1,000-fold. So although there's only a few steps there between those two organs, you have to now explain how the brain can cope with that extra information, how the wiring from the eye can deal with all of that. And that's something we're doing at the moment. Okay, so the other thing that Darwin wrote in the book is he gave us clues about how he was thinking about colour, and that's not very highly. He thought that the eye was this great sophisticated organ, and that colour was just something that does its best to try to adapt somehow. Just before The Origin of Species, he published an essay which talked about an unfortunate case of colour in nature, from which he made a generalisation that colour was the correlation in the visual system. Here he wrote, the nature or condition of certain structures has been thought by some naturalists to be no use to the possessor. So colour was malfunctional. But to have been formed wholly for the good of other species. So for example, fish are certain colours to aid the predators to catch it. He also said, no doubt one being takes advantage of qualities in another and may even cause its extermination. But this is far from proving that quality was produced for such an end. And the bright colours from fish might result from the exposure to certain chemicals in the environment. So really he was saying that the colour, it's not adapted, but it's not the fish's fault. It's just incidentally absorbed certain colours, certain chemicals from its environment that have made it that colour. But that's really the wrong way of looking at things. I mean, he was talking about a fish in water actually that had turned brown. And this actually stood out within the water because he was examining the fish in his laboratory. But when you look at this fish in the water, in its natural environment, the water actually has a reddish-brown colour, and the fish disappears from sight. It's actually perfectly camouflaged. So Darwin really missed out on that, and that's something that this talk will address. So I want to show that colour is not simply a byproduct of its environment. Almost every time we see colour in nature, it's adapted. It's really well adapted to its precise local environment. And in fact, it's extremely sophisticated in many cases. It's not just the correlation, it's as sophisticated or even more so than the eye itself. And in fact, colour often outwits the eye. There's a whole arms race going on between colour and vision, and sometimes colour comes out better. But I think what I want to really get across is the sophistication of colour in nature. And colour is alive. It involves electrons jumping around within atoms. It involves, or even between atoms, within molecules. It involves light rays bouncing between little parts within a structure. The whole system is alive. And eventually the eye gets to see what is actually left over from this process, what's reflected back, and the eye detects that. But before it gets to that point, it's gone through a whole series of sophisticated movements. And the whole thing is an energetic system that's totally alive. And so so light, as I said, colour doesn't exist, of course it doesn't. And this sort of indicates, I work in both biology and physics laboratories, and the picture at the bottom shows the difference between the two there. In biology, we'd be considering that picture of flowers, and in the physics laboratory, we've got this graph showing the wavelengths that are reflected from each of those. And so, two very, very different ways of looking at the same picture. So this colour, of course, doesn't occur until somewhere between the eye and the brain. That's where actually colour is filled in. And called the ways of producing colours factories, because they are these sort of little engineering devices, these things that we've got things, moving parts all over the place. And I've identified really 7 different ways that colour is produced in nature. Okay, we'll start off with bioluminescence. Now, I'll follow the theme of the book actually, but that's not so important here. The theme is that each chapter has a separate colour and is talking about a separate mechanism for producing colour. And I'll start each chapter actually with a question, which is interesting in this case. It was when I received a letter from the Philippines Department of Fisheries, because they had a problem. There was one local town on the coast, and the people were leaving their houses. Everybody was moving and deserting the place because they believed there were some spirits in the sea. Every so often, the whole bay would light up a luminescent blue, so bright that it could be seen from the moon. And the locals, the only explanation they had was this was some kind of spirit, and this spread became so serious that the locals were leaving their houses. So I was called in to investigate that one. And as you would expect, this is obviously a case of bioluminescence. So bioluminescence is common in nature, particularly in the deep sea. Below about 200 metres, over 90% of all animals bioluminesce. And it's often found at night in caves on land. So it involves a chemical reaction. It's where a luciferin and a luciferase react in the presence of oxygen, and light is produced as a byproduct. And it's often called cold light. It's a very efficient way of producing light. Nearly all the energy is converted to light, compared to, say, less than 10% in a conventional light bulb. Here are some examples. Lots of bacteria produce bioluminescence. So that photograph on the left is bacteria on a petri dish. But this is photographed with no light source. So the light is being produced purely by the bacteria. And here is a fish. You can just about see the eye there, a little bioluminescent organ. And in this case, the fish cultures the bacteria. And the bacteria produces the light, but it also has a shutter that overlies the bacteria culture. So by opening and closing the shutter, you can send out signals, and these fishes actually communicate and talk to each other using those light signals. These are the animals I work on. They're called sea shrimps, or ostracods. They're only about the size of a tomato seed, normally. You can see the one on the left, it's producing bioluminescence, and if you take away that background light, you can see that that's all that you get to see in the water. And they use that bioluminescence for different reasons, actually. To begin with, they used to produce it as a way of startling the predator. So if a fish came to eat the animal, it would flash its bioluminescent light and stun the predator, allowing the ostracod a few seconds to escape. Then they started to get together and produce a cloud of bioluminescence. So in some parts of the world now, at the surface of the sea, they gather and produce a large cloud of bioluminescence. And this acts as a burglar alarm response, because a smaller fish, which feeds on these animals, it's not safe to move into the light, because they themselves will light up as a silhouette to larger fish. So the predators stay away from this cloud of light. And then the third function is something that happens in the Caribbean Sea only. And each species, there are over 50 species there, and they all look very similar. And the way they find their own species for mating is that they have their own specific flash pattern of light. So they come out of the sand, just as the sun goes down in the Caribbean, the whole reef begins to light up. The male comes out of the sand and produces its own specific flash pattern. Some go vertically like this, some start at the top and move down, some all directions. But there are 50 different patterns in total. And we can even prove that because we can take a light down into the sea and flash a specific pattern, and we get exactly that species from that pattern of light. So it works really well. And here's moving on to some of the most spectacular coloured animals of all, actually, the cephalopods. We've got, here these are squid octopus and cuttlefish. And the red colour here is the body of the animal. And the blue and green is the bioluminescence. So you can see the patterns of bioluminescence produced in the water. Some are producing flashes, in some parts of the body are lighting up themselves. The one on the left is interesting, this one here, because it has one very long arm, one long tentacle that it drapes down into the water. And it has a whole series of bioluminescent organs all the way up. But it doesn't flash them together like that. It flashes them one at a time, in sequence. So you start at the bottom and it flashes moving upwards. So when you can't see the body of the animal in the deep sea, all a predatory fish sees is a flash of light moving like that. And it looks like a small crustacean, a small shrimp, flashing its light. So the fish follows it and ends up all the way to the mouth of the animal and gets eaten. But as I said, sometimes colour is as sophisticated as the eye. And here's a typical bioluminescent organ, a photophore. This one is a crustacean. So this is a cross-section, and you can see all the various parts there to it. You start at the base with black pigment to absorb any stray light that passes back towards the body. Above that you've got a reflector, just as in a car headlight. Above that you've got the layer of bioluminescent cells, that in this case make their own bioluminescent chemicals. And then on top of that you've got a lens, a careful system of lenses to focus the light out into a narrow beam. So this is a really sophisticated way of producing a really focused beam, narrow beam of light in the water. And this is an interesting one with a hatchetfish, where it shines its light down into the water column. Right, between about 200 metres and 1,000 metres depth in the sea, sunlight still exists, but it's exclusively blue. All the other colours have been filtered out by then. First of all, near the surface, the ultraviolets and reds and violets begin to become filtered. Then the yellows and oranges, until you're left with blue-green, and then eventually just blue. So these fishes live between 200 and 1,000 metres, where there's blue light streaming downwards. As I'll explain later, some fishes have silvery skin to camouflage themselves, but one problem left is that this fish produces a silhouette from below. So if a predator is swimming below that fish, it detects the blue light from the sky, and then there's nothing, and then it detects the blue light again. So when the light is blocked out, it knows there's a fish above it to eat. So this fish, it has a bioluminescent light and a series of silvery tubes, and it shines its light down those tubes, and they exit in a very focused beam, the same shape as the fish below its body. And it's the same colour as the sunlight, almost. Now, the eyes can detect the level of light shining down to its head, and it adjusts its light so it shines down exactly those same levels below it. So now, any fish, predator swimming below it, sees the blue sunlight, then the blue bioluminescent light, then the blue sunlight again, and effectively the fish disappears. So it acts as a total camouflage mechanism for the fish, and it works extremely well. And we know that because it's evolved independently in different fishes. So there's in the sea, sunlight is exclusive. Bioluminescence tends to be mainly blue, and that's because eyes in the sea are best at detecting blue light. Well, below 200 metres there's only blue light there from sunlight anyway. But on top of that, blue light penetrates water best, even in shallow waters. So eyes in the sea tend to be adapted to blue light, so it's no surprise that the bioluminescence tends to be blue. On land it tends to be green or yellow, very occasionally red. In caves, it's quite often blue as well. We don't know the reason for that, but this is an extraordinary case of the Waitomo caves in New Zealand, and there's lots of other examples around the world as well. Lots of good examples in Australia, I know. This is amazing. You walk into the cave, and when your eyes adjust to the low light levels, you begin to see what appear to be stars all over the cave ceiling. As you can see here. And this is a case of bioluminescence again. If we turn on the lights, we can actually see what's really there. It's a fungus gnat larva. You can just see it in the top here. And it's living within a transparent tube. It makes a network of transparent tubes that, and it lives in one of them, but they're all connected. And they're effectively fibre optics. What it also does is it produces some of the tubes that hang downwards. And over those tubes it secretes a sticky solution, a transparent solution, of the same refractive index material. So the sticky beads have the same refractive index as the tubes themselves. And these beads, as you can see in this image, they form these sort of teardrops as they drip down. Now what happens is the larvae has a bioluminescent organ. When that lights up, light travels through this system of tubes or fibre optics, and it goes down these vertical tubes until it meets an area where there's a droplet. And then you've got the right angles for the light to escape. So while it's in the fibre optics, it's moving by total internal reflection. But when it meets a droplet, it has the angles to escape. So these droplets effectively are the areas where the light is coming from. So they appear like little light sources. So when you turn the lights off, all you see are the drops, the droplets glowing blue. And it's a really efficient system, and it attracts flying insects. So the droplets are also sticky. When the flying insects are attracted to those lights, they get caught in the sticky solution. So a really good way of catching prey. But probably the best known bioluminescent animals are the fireflies. And these are absolutely incredible because the precision with which they can flash their luminous abdomens is quite incredible. It's really, really precise. And again, just like with sea shrimps, each species has its own particular flash patterns. In Southeast Asia, you get examples such as in this tree here, where the whole tree lights up and it flashes, the whole lot flashes in unison. And it looks, you think it must be light bulbs because you've just got these trees just flashing on and off. They're really quite spectacular. But you get these in Europe as well, and when I go to Italy in about May time, that's when the courtship is taking place, and they use this to attract, the males attract a female. And I go out into the field with a little LED light, green light, and I flash it, and I'll see another light will flash back from the other side of the field, and I'll keep flashing it, and the light will move towards me, and eventually the beetle will be sitting on my hand. So it's amazing. It works really, really well. But like the ocean, this is a really good way of how different species can inhabit the same area, because they produce their own displays of flash patterns of light. And this is showing 6 different species there, and the yellow is their bioluminescent flashes, and the blue is their flight pattern. And so all you see in the dark, obviously, is the flashes, but you can see each one has its own pattern of light, which the females can recognise. There's also a crafty predator related to these beetles, and that actually mimics the patterns of flashes of the one. So it notices what flashes are actually being used in the area, and it will mimic the same pattern. And so when the female comes towards it, it eats it. But it's better than that. It also copies exactly the height, the flight height of that beetle and the flight pattern as well. It's really incredible. So going back to the original question, what caused that light in the Philippines? Here's a picture of the bay, photographed from a boat, and you can see the people on the back of the boat with the engine of the boat there. It really does light up incredibly bright, and you can understand why people were a little frightened. But in the daytime, the bay looks like this. What's happening is that there are organisms called dinoflagellates that are produced by luminescence, that are microorganisms in the sea, and they get washed into this bay. Now they produce their bioluminescence when they become agitated. When they get washed into the bay, the water keeps coming in and agitating them, so they all together, in very large numbers, produce this really intense light. The cumulative effect is really quite brilliant. So here are those animals, and they do, there's up to 720,000 individuals per gallon there. So this chapter, or this colour chapter that I've been talking about, bioluminescence, it gives lots of indications about how sophisticated colour is, and also the fact that the eye isn't perfect after all. So, for example, here we've got the ostracods that were producing a cloud of luminescence that was stunning a fish predator. It was overriding its visual system, and it had to wait a few seconds to correct itself, by which time the ostracod had gone. So colour wins in that particular arms race. And you've got the fish that are deceived by the bioluminescent light being shone down on the other fish. So quite often colour is the winner in the visual arms race. The mechanisms, as you've seen in some of these cases, are really quite sophisticated. On a similar theme, actually, there's this interesting case of animals in the deep sea that are just going missing. If you took a camera in the deep, you would actually see red animals just suddenly disappearing from sight. Well, because we're talking about the area where sunlight is exclusively blue in the sea, and you find a lot of animals are red to be camouflaged, because red is something that doesn't light up under a blue light. It absorbs all of the blue. So these animals should be totally camouflaged under bioluminescent or light or sunlight. But yes, some of these things are being detected by eyes. Predators are somehow coming along and eating these animals that should be totally, according to our theory, camouflaged. But then we came up against an answer in the dragonfish. All of a sudden, in the 1990s, these species were discovered. And in a world where everything is blue, these species are producing red light. So everything is adapted to blue, being red to be camouflaged. And then the dragonfishes have a light organ that's producing red bioluminescent light. So it has beams of light shining through the water. And on top of that, it's got an additional red pigment in the eye that's believed to actually derive from an algae from its gut. So it's going along shining red light and seeing it and lighting up 90% of the animals in the sea without them having an idea of what's going on. Here's an example of the visual pigments in the eyes in deep sea animals. As I mentioned, you can see most of those are focused around the blue region. Interestingly, they're at the blue region of bioluminescence and not the sunlight. You can see the sunlight is just to the left of that there. But then, yeah, we've got these strange things suddenly with a red visual pigment. So here they are, lighting everything up, and no fish has any idea what's going on, other than that it's being eaten. So again, the eyes of these deep sea fish are certainly not perfect because they can't see the predators approaching at all. Moving on to probably the commonest form of producing colour in nature, which is pigments. of level of sophistication of colour in nature. And most people just think of pigment and they think it's quite dull and boring. There's not much to it and there's not really a subject there at all. But even colour pigments, there's quite a lot going on with those. And in this chapter, the one interesting question is why do kestrels hover above motorway verges? So at least in the UK, when you're driving on motorways, you see these birds of prey hovering alongside the road. And you think, why are they there? I mean, you'd expect their prey maybe to be camouflaged in those areas, because quite often these are brownish yellow fields, and the mice that are hunting, the rodents there, are the same colour. And everything looks perfectly camouflaged. So at first sight, the first answer to this must be ultraviolet. Well, here's the sun's solar spectrum of the Earth's surface under 3 different cloud conditions. So you've got no cloud above, and then going down into more and more cloud. And you can see the peak in the sun's output is just at our visual spectrum, between 400 and 700 nanometers. And that's why the eye evolved, to see those wavelengths. It's the optimal wavelengths to detect. But there are other wavelengths as well, even in smaller quantities. There's infrared, which at the moment we don't believe anything's using. There are little signs that some animals might be using infrared, but nothing's been demonstrated yet, not effectively. The one interesting sign was somebody found that when they were turning on their TV using their remote control, their goldfish would turn around in its bowl. So you've got the ultraviolet, the infrared beam there, and the goldfish is reacting to it. But everything has been quite anecdotal. I think there was also a paper that talked about that owls were responsive to an infrared light each time it was turned on. But the problem there is there was probably a sound associated with the turning on of that light that an owl could hear and we couldn't. But yeah, to the left of that 400 nanometers, 0.4 microns there, we've got a reasonable amount of ultraviolet light. So we know what ultraviolet B does really harmful things to DNA and RNA. It denatures them, it bends the molecules in harmful ways. And lots of things have natural sunscreens to deal with that. But here we're talking about UVC, the far end of ultraviolet, that part that's nearest to violet. So here's a photograph of a flower in ordinary visible light and then in ultraviolet only. So we wouldn't see this picture on the right-hand side. We don't detect it because our retinal cells would detect ultraviolet, but our lens and cornea is not transparent to it, it absorbs it. But here's a fairly ordinary picture. The patterns can become more ornate. They can have, flowers tend to have really ornate patterns, and what we believe are landing strips for insects, because lots of other animals do detect ultraviolet light. In fact, most do. Particularly insects and birds. So, on top of what we see, there's a whole world going on all around our eyes that we just don't know anything about. There are really nice patterns in animals and plants that we just don't know are there. We're totally oblivious to. And we can pick these up, of course, using a camera with an ultraviolet-only filter. We used to have to use special film, but now most digital cameras can pick them up quite easily. Because although in general people talk about glass absorbing ultraviolet light, it's only the real end of the ultraviolet spectrum that glass is mainly transparent to. So here's the absorption spectrum in the eyes of a few animals. And along the bottom there tells you the wavelength. So that's the human spectrum at the bottom. It cuts off at 400 nanometers. But you can see that all those other animals, the bird, gecko, goldfish and butterfly, they've all got an extra pigment that can see in the ultraviolet. And ours would, our blue pigments would actually go that far as well. But pigments are alive with activity as well. Here's a couple of examples. One way that pigments work are by electrons moving around within a molecule. Typically what can happen is that sunlight, white light from the sun, will strike a molecule and provide enough energy to dislodge an electron in an atom around the outside of the molecule. And that electron will then travel between atoms via double and single bonds within the molecule. Now, as it travels around, it releases heat. Heat is generated as part of this process. So the energy in the light is actually being turned into heat and eaten up effectively. Now, some different pigments obviously eat up different wavelengths of light. So in white light, a black pigment will eat up all the different wavelengths, and so there'll be nothing left, and it will generate a lot of heat. And a white pigment will not eat up any of them, and there'll be no heat generated, and everything else will be scattered back into the environment. So we see what is not eaten up by the molecule, but rather what is scattered back out into the environment. And it's the same effect from every direction that you look at it. So with a black pigment, it's eating up all that energy, turning it into heat, and that's why a black t-shirt will feel hot on a summer's day, compared to a white t-shirt which doesn't produce any heat, for example. And then you can go through all the different colours. A red pigment will eat up all the different wavelengths except for the red, and the red will be scattered back out. So pigments obviously are the commonest cause of colour in nature. In terms of evolution, we try to look at how they evolve. They always start out as a byproduct of the physiology of the animal. So here's a marine worm, a polychaete worm, that has been pulled out of its tube. Now it normally lives in a very camouflaged, opaque tube. And when you pull the worm out, it's got colours in there that it's being produced as part of its physiology. But of course it's not adapted to the environment. If this crawled out of its tube, it would be quickly eaten by a fish predator, because this is a nice chunk of protein just waiting to be eaten. So evolution would start to result in the evolution of camouflage or warning colours, for example. Natural selection would act on it, because there are eyes everywhere in the environment around it looking at it. It's leaving an image on retinas everywhere, which becomes a selection pressure for this to evolve, and to evolve camouflage colours so it can't be seen, or bright warning colours to warn of a toxin, for example. Going back to our mouse. Well, we thought that, to us this mouse is brown, but still these birds of prey are seeing it. They have really good eyes. They have really good telescopic spots on their eyes that they can magnify certain things. And we thought, well, okay, the mouse must have an ultraviolet content that's not adapted to its environment, and that the bird of prey is seeing. But it didn't. When we looked at the spectrum of it, it was mainly red and a little bit of white, producing the brown effect, really. But then we looked at the trails of the vole, and the vole was leaving trails of urine that strongly reflected the ultraviolet. And what those birds of prey are doing, those kestrels, they are finding some of these markers, some of these trails, by the ultraviolet reflection, and they're hovering over them. And this represents a pathway for the mice. So they wait there, waiting for an actual mouse itself to come running past, which they detect by its movement, and then the mouse is eaten. So again, lots of examples of why the eye is not perfect. The human eye, of course, is not perfect because it can't see ultraviolet. This whole world all the way around us. The mouse eye isn't perfect because it's not adapted to seeing things directly upwards into the sky, and so on. And just while you're on the subject of pigments, a subject we've been talking about this morning already, and that's the fact that the colours are not always what they seem. This is probably the best case because it's the green tree frog of Australia that has the Latin for the word blue in its name. Cerurea, a certain type of greyish blue. And the reason for that is that when this frog was first discovered in the 18th century by the first fleet of boats that went to Australia from the UK, and there was a botanist on board that went out collecting animals, and he collected one of these frogs and stored it in a bottle of rum. They didn't know what else to do. They were just at the moment when rum was being explored as a way of preserving things. Alcohol, sorry. So all he had was a bottle of rum, so he put this frog into it and shipped it back to London, where it stayed for several years, before anyone came to look at it. And when someone did look at it, they pulled the frog out of the bottle and it looked as you can see on the right-hand side. So it's a blue frog. And it was named with the word blue in its name. So what's happened here, obviously, green is a big problem in nature because lots of animals would benefit from being green to camouflage against leaves, because obviously chlorophyll is very common in the world. But green pigments are very difficult to evolve for animals. There's only a few cases, if any, of a green pigment. So to get around that problem, green animals usually result from blue plus yellow. And quite often, blue is a structural colour, which I'll talk about next. It's a physical structure that produces the colour, totally transparent materials, but a physical structure. And the yellow is a pigment. So just as a paintbrush with yellow paint, the paint would wash out in an alcohol. That's exactly what happened in this bottle of rum. The pigments dissolved into the alcohol, leaving us with the blue structural colour. So we're left with the green tree frog with the word blue in its name. Here's an example of what's going on, for example. We could do lots of experiments like this. This is showing how yellow can be produced in different ways. At the top we've got a monochromatic yellow, which is just a single or narrow band of wavelengths of 580 nanometers. And in the bottom, you're producing yellow again from no yellow light at all. So combining green of 540 nanometers and red of 620 nanometers. So what looks to be 500 nanometer yellow is not present at all. So again, the eye is tricked and is far from perfect, and this is what evolution has exploited. Just another interesting point on pigments, they're not always distributed within the skin, but sometimes within cells within the skin, called chromatophores. At the top, we've got a chromatophore, the same cell, photographed expanded and contracted. So these are cells with very elastic cell walls, with several muscles attached to them, and they're under nervous control. They contain pigment. When they are pulled apart by the muscles, they become large, and the pigment becomes visible, so it becomes a pixel, as on the left. When the muscles are relaxed and they contract, the spot becomes so small it's no longer visible to the unaided eye, and you no longer see it. And so this is how things like chameleons and cuttlefish can change colour. You imagine having chromatophores filled with different pigments in the same patch of skin, and they can expand and contract in different ways. So, for example, this doesn't happen. If you have red, blue, and green chromatophores, you can produce any colour that you wanted. Probably the best case of chromatophores is in the master of mimicry, the mimic octopus. But this uses its whole body to disguise itself too. Here you have this amazing animal from Indonesia. It's quite a small octopus, and it has a lot of predators in the seas where it lives. But there's a lot of poisonous animals living there too, a lot of venomous creatures. And it mimics those. And it knows which one to mimic depending on which predator approaches. At the top you can see the normal behaviour of foraging. And then below that it starts to mimic a flatfish, which has a poisonous spine on the top of its spine. It moves its arms around. It also swims with the same undulations along the sea floor as the sole. And then next it's mimicking the lionfish by spreading its arms out and contracting its chromatophore to produce really strong black and white stripes. And the same again with the sea snake mimicry, where it hides in its burrows and just allows two arms to come out and mimic the sea snake. really amazing animal that actually causes us quite a lot of problems to explain in terms of evolution. And here's a view of cephalopod chromatophores. This is the skin of octopus, cuttlefish and squid, showing the different chromatophores. So magnified so you can see the different cells. And you can see in each of those that some are expanded and some are contracted. And so you get the idea of how this mechanism works. It sometimes becomes more sophisticated. Sometimes there are silver reflectors behind them as well that can add another element to this. But these are really amazing things that could have all sorts of uses in the commercial world as well. And the squid use these colours actually to communicate between themselves. Here are some examples of one species producing different patterns that mean different things to their own species. And we can learn the language of this. And actually you can take a computer-generated animal and change the colour of it and get the other animals in the water to do exactly what you want. It's complicated how this mechanism works. It's all driven by the eye. Quite often it's used for camouflage in these animals. And the eye makes an assessment of what's the best colours and patterns to copy. And that information from the eye is sent to the optic lobe, which sends information to the lateral basal lobe of the brain, which sends information to the posterior or anterior chromatophore lobes of the brain, which then sends the information to the skin. And although it looks like it can do almost anything, there are actually only around 20 different patterns possible from one animal. So you've almost got like a switchboard in the brain, and this information is kind of plugging in a certain key in the switchboard to change to a specific pattern. But they mimic really quite incredibly well. And this is the blue-ringed octopus, which although the blue rings light up at certain times, when the animal becomes aggressive, the blue rings appear to light up. In fact, they don't. It's the background that changes. It has a blue structural colour that's really bright until you put the white behind it. Because then the blue is reflected, then all the other wavelengths pass through and are back reflected by the white layer. That white layer, however, has black chromatophores within it, and when they expand, the white layer becomes black. So then the blue light is reflected and all the other wavelengths are absorbed, so the blue light stands out like a strong electric blue colour. The other interesting thing is about camouflage, because there was one cuttlefish that appeared to be camouflaged on the sea floor in shallow waters, in areas where dolphins live. And it turned out dolphins were the main predator. And when we looked at it, we were puzzled because it stands out extremely well. It was stationary on the sea floor, as if it's totally camouflaged. And we wondered what it was doing, because it's so obvious. And it turned out that if you photographed it in black and white, or effectively mimicking the dolphin's vision, it became totally camouflaged. So it's part of this arms race that intimately involves the dolphin as a predator. And then finally, I've just got this photo here to show that pigment, that pigment probably drove the evolution of colour vision in humans, in the human line, because, in primates, it's believed to be able to, colour vision can pick out the difference between ripe and fruit. So without the red receptors in our eye, we wouldn't be able to pick out the difference in that top picture between the ripe orangish red fruit and the unripe greenish fruit. We would see a picture below where everything looks equal. Okay, the chapter on yellow is something, it's a new subject really for biology. It's something well known in geology, but for a while ago, I think in 1922, I think possibly in Germany, there was a collection of parrots that someone realised under a fluorescent light, some parts of the parrot started to glow. And this was just sort of dismissed as a byproduct of something at the time. wasn't really considered very important. And it's only very recently that this has been taken up again and found to be important. So this is a budgerigard, photographed under a fluorescent light only. In other words, there's no yellow light in the illumination, but this parrot is glowing yellow. Here you can see again. What actually became important, and what we certainly noticed was important, was that not all of the yellow areas were glowing. First of all, it was being dismissed as this is obviously fluorescence. This fluorescence is a byproduct of the yellow pigment. So all the yellow feathers were just incidentally fluorescing as well. But you can see now that it isn't, that's not the case. As you can see, the lower part of the chin there, some of those yellow feathers are fluorescing, and some are not. And also the stripes on the back of the head. Here's a case with another parrot. And here's a case of Indian art as well, where a natural fluorescence was supposedly involved here. The one way to tell if a piece of Indian art is genuine is to shine a black light on it, a light with a high ultraviolet content, because the yellow will suddenly glow, and it glows really, really strongly. It's a really pronounced effect. And if you took a spectrometer and you looked at the yellow pigment on an Indian painting before 1900, then you'll get over 100% in the yellow region, which is a bit odd. So that yellow in that painting is reflecting with over 100% reflectivity. So how can it reflect more light than it's actually illuminating it? So the answer, of course, is fluorescence, and the yellow pigment actually was derived from the urine of a cow. The cow fed with mango leaves produced urine. It's kind of a well-known story, and but it was banned in the 1900s because the mango leaves have no nutritional content for the cow, so it was considered cruel. But what's involved is fluorescence, and fluorescence has been long known in the world of mineralogy. It's a way to actually identify certain minerals by their fluorescent emission. So you've probably seen in the museum specimens where you press a light and the ultraviolet light comes on, black light, and the specimens start to glow different colours. And that's where the phenomenon was first discovered. And fluorescence involves, again, the movement of an electron. So if we simplify an atom, we give an electron an orbit. Ultraviolet light comes in, which is high energy. It causes an electron to jump into a new outer shell, the outermost electron. But instantly, that electron drops back down to its original shell. Now what happens in that process is that heat is given off. So some of the energy is lost as heat as the electron makes its move up and down. So we have to remove some of the energy from that original incident light in the output light. So we go from high energy to less energy light. We go from, in this case, violet, ultraviolet, to green. So ultraviolet light comes in, which we don't see, and green light is emitted, which we do. It was also known in scorpions as which I think it's a byproduct actually of a urine product that covers its body. But scorpions are well known to glow under an ultraviolet light. But probably this has no biological function. It's probably incidental because it's not actually observed in the wild, only under laboratory conditions.
This is the way you actually in nature.
It is, that's right, yes, yeah. No, I work with the BBC have been borrowing my ultraviolet lights to go and catch them for their documentaries. And it works extremely well. You walk around the desert with an ultraviolet light, and suddenly all these scorpions light up which you don't otherwise see. And of course, fluorescence is known in all sorts of animals, our teeth, fluoresce incidentally. Here's a case of an anemone under white light and ultraviolet only, showing only the fluorescence. More examples. This one's interesting because this is a coral. that's fluorescing. What's interesting about that is in the top picture, that's what we would normally see if we were diving, and if we were doing a survey of the sea floor to map the biological diversity of the sea floor. But it's really hard to pick out what's there. But under ultraviolet light, everything has a different fluorescent emission. So this is unnatural light now shining on it, but you can pick out clearly what's there, and you can identify things much more easily. Here's another example. There's a shrimp on the top, which is very difficult to see, but by its fluorescent emission, it begins to stand out. So you can crack the camouflage of many species. That's turning out quite useful. And this is just to show the different emissions of two different species that are otherwise difficult to separate. Also, more importantly, we've been doing remote sensing, and fluorescence is an indication of photosynthetic activity. So rather than just trying to map the distribution of photosynthetic organisms in the ocean, which are the things responsible for oxygenating the atmosphere more than anything else, we can actually map now their productivity, which is more important than abundance. So the more fluorescence, the more productivity we produce. So we know, for example, the best parts of the oceans to protect. Here we're just starting to build a map. You can just see India there. We've started to fill in those areas of the sea. And you can see some are more productive areas than others in producing oxygen. And then some people have been playing around with the fluorescent gene. So they take the genes that are fluorescence. Obviously, the gene of fluorescence in jellyfish is very useful in the medical world, you all know. But they've been putting it into all sorts of other animals as demonstrations that you can actually, if you put the gene that translates the fluorescent protein into almost anything, it will be expressed and fluorescence will occur. And there it is in some fish. I think a rabbit's been done as well. This is my favourite case. This is great. This is a stomatopod, a mantis shrimp. Probably the most visual animals of all. I think they have 16 visual pigments in their eyes. And they use fluorescence in areas where there's only blue sunlight, and they can appear yellow. So where everything else is only blue, it has a pigment that's fueled by blue light, and it emits a yellow light. So in the sea, where it's only blue light, you've got these yellow animals appearing. And they, of course, can detect that yellow themselves. Okay, now this is really the subject that I do most of my research on, which is structural colour. And in this chapter, which is the violet chapter, it talks about there was this problem because there's this bird, this butterfly, as you can see here, in Southeast Asia, that's a very bright violet colour, and it's flying around in the middle of a whole group of different bird predators, flying around quite happily and not being eaten. It has no toxin, it's not distasteful to the birds. And we wondered what's going on, why is it doing that? Anyway, structural colours are, as they say, they're physical structures that produce colour. So there's no pigment, usually totally transparent materials that interact with light rays in different ways, with lots of reflections and at different intervals within the structures. So you've got light rays bouncing around within these devices. Typically we have dimensions on the order of 100 nanometers. As you can see with this butterfly, typically the colour changes with viewing angle. So there, the green part of that butterfly in the centre, as it turns around, when you change your direction, becomes blue. Probably the first person to write about that was Newton in optics in 1704, when he guessed really that the peacock feathers must have some kind of structural colour within them, some kind of prismatic effect. There aren't any prisms in nature that are used for producing colour, but that's probably a good example of a structural colour. It's a physical structure splitting up white light into a spectrum. And we have all different manners of structures out there. that are working in slightly different ways to produce the colour. At the simplest scale, we've probably got scattering. Just like the pigments scatter light back out, we have little particles such as in the globules in milk that scatter white light. Basically, we scatter all wavelengths in white light equally and randomly into all directions. So they reform to produce a white effect on the retina. However, when particle sizes become smaller than the wavelength of about yellow light, they start to reflect more of the shorter wavelengths blues than the longer wavelengths red. And we get something called teal scattering, which is now not as common as we thought it was. This is two animals that have got teal scattering because one's eating the other. Very transparent animals, and the one's taking in the small particles from the other, and so it's starting to scatter light too. And in its simplest form, teal scattering is all about the particles actually making contact with some wavelengths and not others. And at the bottom you can see what's going on there. You can see the long wavelengths literally miss the particles, where the shorter wavelengths are more likely to hit them. So more of the blue light is reflected, the more of the red light passes through. And that would explain why the sky is blue. The particles in the sky scatter the shorter wavelengths and the longer wavelengths pass straight through. So when you're looking at a sunset, you're looking at the wavelengths that are passing straight through the system, which are the red ones. And the blue is actually being scattered by the particles. That's where I just come from. And actually, because these are physical structures, we can still get signs of them in fossils. This is the dodo. This is what we had. This was the last dodo specimen preserved, which we had at Oxford University. And the curator at the time thought it was so scruffy, he decided to throw it away. We thought it wasn't fit for exhibit. Fortunately, somebody saved a head and a leg, and that's all we've got left of this animal now, which has been very useful for DNA studies. But also in the blue area of the beak, we found these tiny structures that would produce the scattering of blue light, which confirms why in some paintings of the dodo, they do indeed have a blue area on the bill. And it's probably true, that bill probably was blue. The main types of metallic, structural colour or metallic colour in nature are single and multiple thin films and diffraction gratings. Diffraction grating is a surface structure that is a series of parallel grooves or bridges that splits up white light into a spectrum, just like on a CD or a hologram of a credit card. I started working on seed shrimps. We've got an example, just this little guy there. And these are examples from traps we were setting in Australia, down the east coast of Australia. And we were catching sometimes the animals became quite big, as you can see on the bottom there. This is an animal that's related to a wood louse, a little crustacean that you can get in houses quite in damp areas. And normally they're only about that big, but these were this big. So it was quite amazing with strong jaws as well. So keep out of their way. But I worked on ostracods as seed shrimps, and this is what they look like. These ones were nicknamed Beth Beans. But on their antenna, they have the diffraction grating, as you can see there, and they're producing these really nice iridescent colours. And when I filmed two individuals in courtship, they were using the light as a courtship display. So normally they have shrimp-like bodies, totally enclosed within a bivalve shell. When the female gets within viewing distance of the male releases these iridescent hairs from outside the shell, and the female becomes receptive and mating takes place, as these frames on video recording show. And I looked at the evolution of this group, and it's a long story, but effectively what was going on is that over 350 million years, the diffraction gratings are becoming more efficient, more optically efficient, producing a very bright blue light in the water. So these animals are evolving with light as the major stimulus. to the point where in Australia at least they become the commonest animals in the sea. So a very successful mechanism. And diffraction ratings weren't known in nature before, so I started to look to see where else they occurred. And they occur in lots of other animals. Once you start to get your eye in, you can start to find these in a whole range of forms as well. And this was an interesting one because I was looking for these gratings everywhere, and I found this one in Baltic amber. And this was some work that in Germany in the 1970s, somebody took a fly from Baltic amber that was 45 million years old and revealed this micrograph. And the micrograph shows 4 facets of the compound eye, and you can see how well it's preserved in amber. And you can see these fine striations on the eye, just there. And I was wondering what they were, so I reconstructed that in a photoresist material using laser beams. And here are the graphs below to show the response. A smooth surface of the material reflects about 5% of the light over a whole range of angles, and then the reflection got bigger and bigger. So just like a glass window, you see a 5% reflection, and you see a reflection of yourself. But the fly's eye surface didn't reflect anything. It was down to below 0.1% over a range of angles to 60 degrees either side of the normal. So if you put that onto a glass window now, you no longer see a reflection. This was great for the fly because it was allowing all the light to pass through the surface, so it could enhance the vision. But it's being used now on solar panels. We've been making this with the Fraunhofer Institute for Solar Energy Systems, and we get a 10% increase in energy capture from solar panels by employing this, because now there is no light lost from reflection, and it's all going through to the photosensitive cells and being absorbed, turned into energy. And now the thin films reflectors are more common. An example is a fly's wing or a layer of oil on water. You've got effectively 2 layers here. We've got material of different refractive index. So you've got a layer of material there, and it's got an upper surface and a lower surface. So rays come in, some reflect on the upper surface, the rest passes through, some reflect on the lower surface, and the rest passes through. Those 2 reflected rays can superimpose constructively or not. And if they superimpose constructively, you see a colour corresponding to that wavelength. This makes all the difference how long it's travelled. And if light comes in from a different angle, it will travel further through the film, and therefore it will superimpose constructively for a different wavelength, so you'll see a different colour. So if you view the same structure from different angles, you'll see different colours. A single thin film will reflect about 10% of the incident light, about 5% from each surface. But if you have multiple layers stacked on top of each other, then you have to make sure the refractive index is different in between, so that light effectively recognises that boundaries. With about 10 layers, you'll get about 100% reflection. So these are really efficient reflectors in nature. There's a picture of a sapphire copepod. It's only about 4 millimeters long, but it's really obvious even when you're swimming. And you can see the reflector in this case is within a cell. Usually they're extracellular. The cells usually secrete the materials through 2 layers, and they build up that way. So on the left, you've got the type of reflector we were just talking about, reflecting one wavelength or colour at each angle. And then on the right, you've got different thicknesses of layers. So each one, for one single angle or direction, is reflecting different wavelengths. And effectively, you get all wavelengths reflected, which means you get a silver reflection. And that's what you get with fish skin. So here's a cross-section through fish skin, and you can see the different platelets, which actually move around this animal. There are different thicknesses piled on top of each other, each responsible for a different wavelength or colour, if you like. So you get all wavelengths being reflected, so they add up to white. If you look at a very reflective form of white, it becomes silver. And this is why fish skin is silver. It usually acts as a mirror to make itself invisible. So if you have a mirror in the water, a predator looking at the fish from below sees only a reflection of the surface, so the fish effectively disappears. And this is evolved several times, so it must work. But yeah, as I said, we get multiple structural colours in fossils as well, and lots of cases of multi-layers. This is from the famous Messel hits in Germany. We've got a 50 million year old beetle there, almost 50 million. And this is the head region. You can see its jaws, its head, and the thorax. And the original colours are preserved. Because, these things are physical structures, just like the head is a physical structure. If the preservation is fine enough, then you'll get the structural colours preserved too. Here's another one that we looked at to see what was causing that. And here's a section, and you can see the different layers here from above of the multi-layer reflector. Things got more interesting when we found photonic crystals in nature. Because only in the 1980s, the concept of a photonic crystal was conceived within physics. And in the 1990s, I started to find these things in animals, and at the time I didn't know what they were. Here's a case of a photonic crystal is something that's involves a photonic band gap. I won't go back into that too much now, but effectively, the structures we were looking at before, for each ray, we were getting one reflection. Whereas here, typically, there are multiple reflections. They're very complex three-dimensional systems. These spheres are about half a micron in diameter. So this is the structure that causes the colouring opal. But when you've got all these multiple rays bouncing around many times, it's quite difficult to calculate the interactions and how the light is going to emerge from the system. And then exactly the same thing turned up in a weevil. You can see there. If you take the turquoise areas of that weevil, they turn out to be scales, and then if you chop through those, they're filled with opal, exactly the same structure. So we're trying to work out actually how the animals make these, because these are really commercial products that we could try to make. Animals are made at room temperatures and pressures, whereas in industry they have to be made of really energetically costly processes. They start to get really complex. This is a marine worm, Aphrodite, that has these iridescent scales and hairs. And if you chop through one of those, it's filled with these effectively nanotubes, each one with a different structure to it. And that's what's causing the colour in that original blue violet butterfly. It was a photonic crystal, in this case inverse opal, as you can see here. Instead of having close-packed spheres, it has close-packed arrays of air spaces. Why was nothing eating it? Well, because there is also a model. The animals, the butterfly we were looking at was actually a mimicry ring. So there was another animal going around that looked almost the same. But this one actually was distasteful to birds. So any bird that takes that one learns not to take that coloured animal again, and so the mimic benefits. I mean, it's not a perfect mimic, if you can see between those, the one on the bottom left and the top right. If the bird predator could look at the white spots, he could quite easily pick out the difference between those two, or the wing shape, but it's mesmerised by the bright violet flash, and can't pick out that animal anymore. So again, the eyes of this predatory bird are not perfect. And a lot of these things now, these structures have applications in commerce, so we're considering that now, working closely with the industry to try to manufacture them. Just very quickly, here's some of the things we've done, making, in this case, the moth-eye structure. As you can see on the bottom that we made there, the first iodine. We made liquid crystals that we found in beetles. On the right, we've got the mimics. And as you move our mimics with the beetle, they change colour in the same ways and have the same circular polarization properties. The morpho butterfly structure we've been mimicking, as you can see in the top right. In China, I've been trying to replace the chitin of the material with different materials, such as zinc oxide, that are more resilient. Here's a diatom, where we're manufacturing on the bottom there with deep photochemical etching. And then try to find functions for them, such as in sensors, vapor sensors, where they turned out to be extremely good, better than anything else. And just briefly, this has led to other things. Because we're looking at microstructures, quite often these things have alternative effects, other functions as well, such as in repelling water and self-cleaning. So we've got the lotus leaf there that we're trying to turn into paint to self-clean. And then finally, I've been trying to get this across to everybody. Biology is not easy. That light is such a powerful stimulus, and colour is so sophisticated, and so our eyes and the visual system, and they take up such a lot of our physiology. It's such a powerful thing. It separates itself from other senses because light is always present. There's always the stimulus there. Sunlight lights up everything. Most things anywhere on Earth. Whereas, say, if you take sound, if you don't make a sound, you won't be heard. And the same for the other senses. Now, although only 6 of the 37 phyla of animals possess eyes, over 95% of all species on Earth possess eyes. So there are eyes everywhere. So everywhere you go, you're being lit, and there are eyes to detect you. So you're leaving an image on the retina. So you have to be adapted. Every animal has to be visually adapted. Otherwise it won't survive very long. So you've got this massively powerful stimulus. Now the question is, where did that story all begin? You've got this visual arms race, eyes and vision, and large parts of the body, not just the colour, but the behaviour of an animal and the shape of an animal is also partly an adaptation of vision. So where did all that begin? Well, we started to look at fossils, and I've shown some 50 million years old or even more with colour. So that whole arms race must have been going on in that time as well. But how far back can you go? The answer to that, of course, is to the very first eye. And then if you start thinking in those terms, you start thinking about how important that very first eye must have been. Well, here are some fossils, the oldest ones that I found colour in. And these are 508 million years old, fossils from the Burgess Shale in Canada. And we take a look at this one here. The fossil looks like this. The original chitinous material has preserved well in the rock. If we dissolve away some of the spines from the rock using acid, the surface of it will look like this. It's still got remnants of a diffraction grating. And when I reconstructed that and shone light on it, it looked like this. So even 508 million years ago, animals were still producing these bright colours, really efficient colours. Someone made this reconstruction from me. But this is an accurate colour reconstruction of what the animal would have looked like all that time ago. And before that, we knew these animals, these are quite famous animals from the Cambrian period, originally well known from the Burgess Shale, but since we've got better examples in China and Xinjiang. And we had a picture of a community. It was always in black and white. Just to show you where this falls within the evolution timeline, this is the history of Earth and life on Earth. And you can see at that point there, where you've got this Cambrian explosion. Effectively what happened, around 520 million years ago, all 37 phyla of animals existed. A phylum is something with a specific body plan, an internal body plan, so it has a major difference from other things. We're in the chordate phylum, for example. I've just represented 9 phyla there, but you think there are 36, 37. And what happens is all those phyla existed, they had evolved, but they all had soft bodies, like a worm. Because a worm is less energetic. You don't need so much energy to maintain that body. So everything was a soft body form. Then suddenly, for some strange reason, life changed, and everything independently, all those different 37 phyla, independently evolved hard parts, which are very energy expensive. So why did they do that? This was the event called the Cambrian explosion, or the Big Bang of evolution. What caused them to do that? Well, we've been looking at this mostly for environmental factors, whether something changed in the environment, but I think that it was the introduction of vision. When I looked at how far we can take this story back of the visual arms race, I looked for the very first eye and found that it evolved just at the very beginning of this Cambrian explosion event, just before it happened. And I think what's happened is that by introducing vision into the system of life on Earth, in a predator, suddenly you've got a predator there that can see all these other animals that have got soft bodies, which are now effectively chunks of protein just waiting to be eaten. Suddenly you introduce a whole new selection pressure on everything, and everything has to adapt to that. So this is the trilobites that I looked at, and I traced them back, and they go back to the very beginning of the Cambrian explosion of the eyes. Before that, we have proto-trilobites with hand eyes, but just starting to get signs in some of them that the eyes are evolving. But again, these were soft-bodied, and I believe that when they evolved vision, they then evolved the capacity to swim quickly and have hard parts to be able to do that, because there was this new potential food source all around it. And then soon after, we've got this, what Stephen Jay Gould called wonderful life, these amazing animals that evolved, all these incredible armours and armoury, and they also evolved not just spines and strong armoured plates, but also eyes themselves as counter-defences and streamlined bodies for swimming quickly. And everything changed. The whole behaviour and physiology of animals just changed because of the introduction of what is now the most powerful sense on Earth. And just finally, this is what happened with vision. With the other senses on Earth, they evolved gradually. With hearing, you gradually increase the sensitivity of the organ through evolution. The same with smell, for example. You have one hair that's sensitive, then two, then a bunch of hairs, then more, and they gradually become a complex organ with lots of hairs in them for sensing. But with the eye, as we saw in that early sequence right at the beginning, it was evolving gradually to detect the direction of light, and then all of a sudden, in one little step, a lens was introduced, and suddenly an image was cast on a retina. So with the eye, you don't have this gradual increasing sense through time, but you've got it gradually increasing, and there's suddenly a massive leap in efficiency. And that's the type of thing that can trigger an explosion. You suddenly throw vision in, and colour becomes important for the very first time. So, 521 million years ago, this is how the world would have looked with the most sophisticated visual perceptors of the time. And then 1 million years later, it looks like that. So that's what evolution has to deal with. And those are all summarised in these two books. So thank you very much for listening.
Signal the osmological difference between the inside and the outside of the cell. You know much about that?
No, actually that's not one I know about. Actually, I'm not, I don't really look at what happened with the evolution of visual pigments or circadian rhythms and what really happened with how the light receptors evolved before the eye. But that's a really interesting subject, yes. Yes.
Do you know why the lens, or why it disappeared?
We don't. I think it would have been it would have been an incidental, probably originally. It would probably be a genetic mutation that was an accident, and it would have caused. I mean, because we've had this eye, there's probably fluid within this kind of spherical invagination, and a genetic mutation possibly caused some of that fluid to harden within it and actually start to focus light a little, and that conferred an advantage on that animal. So that mutation was actually retained, and then natural selection works on that even more. And the more it becomes hardened, the more it becomes rounded, the better it becomes at focusing light. So the origins of the lens probably start as an accident, I would imagine. Yes.
So do we assume then that although you started with Darwin, you sort of finished with Lamarck?
Yes. Oh no, not Lamarck. No. I wouldn't support the Lamarckian view of evolution. No, I think we end up with Darwin eventually again, because we do actually demonstrate how the eye can evolve by natural selection, which Darwin had a problem with in the beginning.
In the books you mentioned this explosion around 540 million years, and now you mentioned 520, so you're still obtaining precision on those data.
That's right. Actually, the dates, that's the one thing we've been working on in paleontology really over the last decade or so, is trying to fine-tune the dates. We have the dates started out quite rough, and now they're becoming much more defined. Partly because we've been finding now a lot more Cambrian fossils from different localities around the world, and that's helped to fine-tune the dates through carbon dating of these different sites. And actually, that is the problem I had at the beginning. The dates didn't quite add up in my theory to begin, because the first trilobites, although the theory worked in principle, but then when we looked at the dates, the Cambrian explosion was set up just before the trilobites with the eyes. But then, when they became redated, now it becomes just after, so now the whole thing really adds up. So it works even better now. But yeah, we are constantly fine-tuning the dates, but it's kind of, it tends to be a sort of a range that is becoming narrowed now, so I think we're going in the right direction.
So it's actually shorter than 5 million years now, the actual explosion.
Yeah, I mean the Cambrian explosion event, this most dramatic event, it was less than 5 million years. Probably within 1 million years this whole thing took place. Yeah, really. On the geological timescale, that's really next to nothing.
Thank you very much.
Thank you.